Good morning, students. Welcome to online classes of science by RPSGOS schools. Today, we will continue with Module 3, that is a part of reproduction in plants. In this Module 3, we will learn about artificial methods of vegetative propagation. The various methods of vegetative propagation followed artificially are cutting, layering, grafting, and tissue culture. Artificial vegetative propagation is the process of growing plants by applying artificial techniques which include cutting, layering, grafting, suckering, and tissue culturing. This method is more beneficial as it boosts the quality, yield, and disease resistance in plants and thereby plant products. Thus, this method is quite economically viable. Artificial method of vegetative propagation is carried out by humans on the fields and laboratories that involves human intervention. These methods are employed by many farmers and horticulturists to produce healthier crops with more desirable qualities. Advantages of vegetative propagation are that they are quite rapid, easy, and cheap method of plant propagation. They bear flowers and fruits early. The new plants are exact copies of the parent plant. The plant produces a large number of plants in a short time, and the plants like bananas, seedless grapes, rose, pineapples, and dahlias, which do not produce seeds, can be grown vegetatively by these methods. Let's talk about the artificial methods one by one. Stem cutting. A part of a plant, typically a stem with nodes, is cut off and planted. Adventitious roots develop from the cuttings and a new plant forms. Cuttings are sometimes treated with plant hormones before being planted to induce root development. In this method, cutting of a healthy young branch of a plant having leaf buds is planted in the moist soil. Cutting develops roots and grows into a new plant. This method is used to propagate plants like bougainvillea, chameli, rose, and sugarcane. Another method of vegetative propagation followed artificially is layering. In this, the stem of the plant is bent to the ground and covered with soil. After some time, adventitious roots emerge from the plant parts covered with the soil. This attached stem with developing roots is known as a layer. Young stems that can be bent easily without any injury are used in this method of layering. Jasmine and bougainvillea can be propagated this way. In some plants, a modified form of layering known as air layering is employed. A portion of the bark or outermost covering of the stem is removed and covered with moss, which is then taped tightly. Some gardeners also apply rooting hormone. After some time, roots will appear and this portion of the plant can be removed from the parent plant and transplanted into a separate pot. This method is commonly used by gardeners to develop plants like jasmine, vines, and bougainvillea. Another very common method used in horticulture to develop varieties of ornamental plants and fruit trees, is called grafting. Grafting has long been used to produce novel varieties of roses, citrus species, and other plants. In this method, a bud or cutting with buds of one plant called the scion is kept over the cut stem of a rooted plant called the stalk. Two plant species are used in this process of grafting. Part of the stem that is grafted or attached is called scion of the desirable plant, and the rooted plant is called the stalk. Both are cut at an oblique angle, placed in close contact with each other, and are then held together. The vascular systems of the two plants grow and fuse, forming a graft. Matching up these two surfaces as closely as possible is extremely important because these will be holding the plant together. The tissue systems of the cutting become grafted into or integrated with the tissue systems of the base plant over time. The vascular systems of the two plants grow and fuse, forming a graft. After a period of time, the scion starts producing shoots and eventually starts bearing flowers and fruits. Grafting is widely used in viticulture or grape growing and the citrus industry. The care is to be taken that stock has an extensive root system under the soil. Advantages of grafting. Plants, flowers, and fruits of desired quality can be obtained through this process. New varieties can be developed. Plants which cannot be grown by cutting or from seeds can be developed quickly by grafting. Tissue culture or micropropagation. Micropropagation is a method of propagating a large number of plants from a single plant in a short time under laboratory conditions. Tissue culture is a technique in which fragments of plants are cultured and grown in a laboratory. This method allows propagation of rare, endangered species that may be difficult to grow under natural conditions and are economically important or are in demand as disease-free plants. In this process, under sterile conditions, the plant material is placed on a plant tissue culture medium or a nutrient rich medium for the plants that contains all the minerals, vitamins, and hormones required by the plant. The plant part often gives rise to an undifferentiated mass known as callus, from which individual plantlets begin to grow after a period of time. A callus can be defined as an unorganized dividing mass of cells. When the explants are cultured in a proper nutrient rich medium, the callus is obtained. The growth of callus is followed by organ differentiation. The culture is grown on a gel-like medium composed of agar and specific nutrients required for the growth of the cells. These can be separated and are first grown under greenhouse conditions before they are moved to field conditions. The callus is transferred into another nutrient rich medium containing plant hormones. The cells of the callus in this nutrient medium divide and differentiate into root and shoot and form plantlets. The plantlets so formed are grown in pots filled with soil. Asparagus, chrysanthemum, orchids, and many other plants are grown by this method. With this, we end up module 3. We'll join you soon with another module. Till then, take good care of yourself. Thank you.